So we've got male reproductive anatomy. It's made fairly easy for us because we have this mnemonic here, 7-up. Seminiferous tubules in the testes. It's where your Sertoli cells, your Leydig cells are. It's where your spermatogonia, your germ cells, are going to be maturing, primary, secondary spermatocytes into spermatozoa and to spermatids. Your spermatids will pass into the epididymis. And in the epididymis, this is where the vast majority of sperm maturation takes place, okay? That's probably one of the highest yield points you need to know, is that it's in the epididymis. That is where the vast majority of sperm maturation takes place. It takes about 9 weeks, so 63, 64 days. That is the amount of time it takes for sperm maturation, and it occurs in the epididymis. As we discussed before, although the vast majority of sperm maturation occurs in the epididymis, if they ever ask you for where does the final step of sperm maturation occur, some students will get this wrong. They'll select epididymis because that is where most maturation occurs. But the final step occurs, as we said before, in the vagina. Okay, it's capacitance. Once the sperm hits that ferning pattern of the female ovulatory vaginal secretions, its motility increases significantly. It's called capacitance. That's actually the final step of sperm maturation. Okay, so during ejaculation, remember our pudendal nerve, S2, S4, that's somatic for ejaculation. We get this movement from the seminiferous tubules at epididymis, and we're in our vas deferens now, and we're traveling through our vas deferens, also known as ductus deferens, until we get to the ejaculatory ducts. We're in the ejaculatory ducts, and we pass into the urethra. Okay, we've got our prostatic urethra, bulbous urethra, penile urethra. Okay, so we pass through. We're in our penis, and that's it, okay? So that's our pathway for sperm travel, and it's pretty high yield. Now for the flash quiz. So during an ejaculation, sperm in the vas deferens come immediately from where? The epididymis. Remember our 7-up? Okay, so we have our seminiferous tubules, epididymis, and then to ductus deferens, ejaculatory duct, and then we'll go to our urethra and penis. So for our male sexual response, the first high yield point is that erection is parasympathetic, right? And it's the pelvic nerve that regulates that. And the other thing is that nitric oxide and cGMP always go together. Don't mix that up with CAMP. Always have nitric oxide equals cGMP in your mind, because NO increases the activation of guanylate cyclase, which will increase your cGMP concentration. And cGMP is going to activate protein kinase G, which will dephosphorylate and relax your smooth muscle of the penis. So it's increased blood flow through the corpus cavernosa, corpus spongiosum, and bulbospongiosus that contributes to the maintenance of erection. Okay, so you get that increased blood flow in the penis. And the other thing is norepinephrine, which is more associated with the sympathetic nervous system, will lead to the opposite effect. Okay, so under stress situations, an erection is less likely to be maintained. So this is also how erectile dysfunction drugs work, such as sildenafil, which is Viagra. So it's a phosphodiesterase inhibitor that prevents the breakdown of cGMP. So as long as cGMP is high, because we've given that phosphodiesterase inhibitor, sildenafil, we can maintain that vasodilation. So emission, now this is commonly confused with ejaculation, in that ejaculation is actually somatic, okay? So ejaculation is not sympathetic. So you have erection that's parasympathetic. You have ejaculation, which is somatic. Ejaculation is not sympathetic. It's emission that is sympathetic. And emission occurs right before ejaculation, as the sperm are released from the epididymis and seminal fluid flows into the posterior urethra. So that is sympathetically regulated. Now for the flash quiz. So during erection, nitric oxide causes an increase in concentration of which intracellular molecule? cGMP. Okay, recall that nitric oxide upregulates guanylate cyclase, which will increase cGMP, our intracellular molecule responsible for activating protein kinase G, which will dephosphorylate and relax the smooth muscle in the penis, permitting vasodilation and erection. So our seminiferous tubules is where our sperm is produced. Okay, this is the lumen where mature spermatids are going to be formed, but they start spermatids before they become as such. They start as these germ cells on the blood-testes barrier, this basement membrane here, and this is where our spermatogonia are, that are these germ cells. Okay, so these germ cells will move towards this lumen, and as they do that, they become primary spermatocytes, secondary spermatocytes, and then spermatozoa. So those germ cells are replenishing our pool of spermatozoa. And we also have Sertoli cells, which are responding to follicle-stimulating hormone. Okay, so follicle-stimulating hormone will increase aromatase production in the Sertoli cells, and the aromatase is converting the androgens from Leydig cells into estrogens. So and our Sertoli cells will secrete inhibin to negatively feed back on our follicle-stimulating hormone produced from the anterior pituitary. The Sertoli cells are also secreting androgen-binding protein. This androgen-binding protein is maintaining the testosterone concentration locally in the seminiferous tubules. Okay, so you need that threshold, that minimal threshold testosterone concentration to enable spermatogenesis. Okay, so the androgen-binding protein will facilitate that. This is different from sex hormone binding globulin, which is produced by the liver, which allows testosterone to travel in the blood. Okay, so your steroid hormones bind to binding globulins in the blood. So sex hormone binding globulin produced by the liver is different from androgen binding protein. Our androgen binding protein is strictly to maintain a threshold concentration in the seminiferous tubules for effective sperm production. And it's our Sertoli cells that are forming this blood-testis barrier. So if they ask you about the cell that is forming this barrier, Sertoli is your answer. And they're also providing nutrients for these developing spermatozoa. And our Leydig cells, okay, so notice that our Leydig cells are outside this blood-testes barrier. So our Leydig cells, also known as interstitial cells, these are the ones that are responding to luteinizing hormone from the anterior pituitary. And testosterone in turn will negatively feed back at the hypothalamus and anterior pituitary to decrease luteinizing hormone. But most importantly is that notice that they're out in the interstitium. Okay, they're out here. They're not inside here, like the Sertoli cells or the spermatogonia are. Okay, they're outside. They're in the interstitium. If they ever show you histology like this, they might mention that there's cryptorchidism, which would be an undescended testis, and you need to know that testosterone production is unaffected by temperature. And if you have an undescended testis, you can get infertility due to the increased temperature. Your Sertoli cells won't work properly, your sperm won't mature properly, but your testosterone function, your Leydig cell function is fine when you have increased temperature, when you have cryptorchidism, this undescended testis. And if they ever gave you a vignette of cryptorchidism, and then they showed you this picture, and they said which cell would be unaffected, many students might find that confusing, but if you know that the Leydig cells are unaffected by temperature, that this testosterone production is independent of temperature, you know that it's these cells in the interstitium. Okay, so those are your Leydig cells. And once again, these are producing the androgens that will move into the Sertoli cells to be aromatized into estrogens. Okay, so your androstenedione, which will aromatize into estrone, and your testosterone, which will become estradiol.